Hello, everyone. My name is Sidney Sheldon, and I am a certified nutritionist presenting on tonight's topic of epigenetics. And while we cannot change our genes, we can change how they are expressed. So in this evening's webinar, we are going to cover a list of kind of how-tos around how we can alter our genetics in the sense of what is actually being expressed. So about me before we begin, and as people join the webinar this evening, my name is Sidney and I am a certified nutritionist. I own a private practice called Veritas Wellness, and feel free to follow me there. I love to share content in the realm of nutrition. My interest in my practice is quite vast. I do not have a niche, but I love to teach. So I have an interest in sharing foundational nutrition knowledge with all of my clients before we begin diving into the deeper work, be that sports nutrition or digestive concerns or anything else. We always start with foundational nutrition because if anything else fails, we know what to come back to. But in my practice, I also love to work with clients who are interested in attaining goals in the realm of sports nutrition and sports performance. I also work with clients who have digestive concerns of varying needs from ulcerative colitis and Crohn's to IBS and SIBO. I also love to work with clients who are looking to manage stress and overall health through nutrition and wellness, and of course, weight management. So my kind of clinic experience is vast and my interest is vast as well, because once again, I just love to teach. I'm also the co-founder of a workplace wellness program where we go into businesses and teach these types of topics to different types of employees and employers through Vallejo Wellness. And I am an InfoFit educator. I am the co-founder and co-writer of the sports nutrition program that we created through InfoFit called the Certified Sports Performance and Fitness Nutrition Specialist Program. It's a wonderful course. If you have not learned about it yet, have a look through. It's a pretty deep dive into nutrition from a holistic sense, and then applying that for trainers and how you can actually work with nutrition for better gains and for better outcomes for your clients, not only in the short term, but for long-term health outcomes as well. I'm also a teacher at the Institute of Holistic Nutrition. So if you can't tell, teaching is a big part of my life. You can follow me at Veritas Wellness or shoot me an email after this session if you have any questions about today's topic. And let's get started. So the goal of this talk is really to share a little bit about not just genetics, but actually epigenetics. And we're going to give you an overview first about genes and genetics just to set the stage. And then we're going to dive into epigenetics and talk about how epigenetics actually affects us in our day-to-day lives, but also downstream, what this can do for us or affect us later on in life. We will then leave you with some tips and tricks and information for you to learn how to create a gene supportive environment for yourselves, but also for your clients. So let's jump in. All right, so let's set the stage here. We've heard these terms before, maybe in high school, maybe in continuing education programs, but let's talk about genetics, genes, and DNA just for a quick second here. So DNA is going to be something that is made up of many, many, many bases. So the DNA in humans is made up of approximately 3 billion nucleotide bases. And there are 4 fundamental types of these bases that make up all of our DNA. You will not be tested on this, but just to set the stage, they are going to be adenosine, cytosine, guanine, and thymine, commonly just abbreviated as A, C, G, and T. And these are going to set the stage for everything that we see as human beings and even things that we don't see inside of our bodies. So the sequence or the order of these bases is what determines the instructions of life. So depending on how those bases are strung out and how they are laid out and then put together, that will determine how someone looks, their predisposition for certain conditions, whether or not we're looking at a piece of hair, a piece of skin, muscle, you name it, will come down to our DNA and how that is transcribed. Now, with about 3 billion bases to choose from, there are roughly 20,000 different genes that have been isolated. So we still actually haven't figured all of this out. This is a pretty new science, but of all of these bases, some of them will be expressed as genes. Others are just going to be base pairs that don't really do anything for us. Okay, so next thing we need to discuss is the genome. The genome is the whole of an organism's hereditary information that is encoded in DNA. And this includes both the genes and the non-coding sequences of the DNA. So back to the idea of some of these genes or these bases can come together to actually make a gene. So it's made up of many different base pairs. Others will not be codable. They're just going to be those letters that I mentioned before that don't really have an expression. Now, while we all look different and have different backgrounds and different heritages, the human genome is mostly the same in all people. So the differentiation between you and your partner or your friend or someone across the world is really small. The genetic variation accounts for about 0.001% of each person's DNA and contributes to differences in appearance and health. Now, obviously, if you have a relative, they are going to have DNA that is more close to you. But even someone from around the world on the other side of the planet, if we were to, let's say, just analyze their DNA, most of it would be very, very similar. But this still leaves room for error, not because of the genes themselves, but how they are expressed. So what are genes? Genes are specific sequences of bases that provide the body with instructions on how to make important proteins and complex molecules that can trigger biological actions to carry out life's functions. So in a nutshell, this is like your blueprint. This is the book of you, your code book. And when we have this code book in hand, it tells our body how to create the cells necessary to function. So cells are fundamental working units of every single human being. We've got tons and tons and tons and tons of cells that do varying things throughout the body. And all the interactions required to direct the activities are contained within the chemical deoxynucleic acid, also known as DNA. So we get the instructions from our guidebook, our blueprint, and then that is transcribed, and we then create the necessary proteins and the necessary molecules from that code. So when we think about genetics, we've heard this in the past of my genes are my destiny. It runs in my family, so I'm going to get it too. Or there's a history of heart disease in my family. There's diabetes that runs in my family. There's Alzheimer's that runs in my family. My family's always been overweight, so therefore I'm going to be overweight too. But let's break that down and actually demystify that. So to do that, we have to make the differentiation between genetics and epigenetics. So genetics is the study of genes that are inherited from your parents. You will receive your genetic code, your blueprint, based on your parents. You will get half from your parent, your mom, and half from your dad. And while the determinants of that are beyond what we're going to discuss in this course, some of the genes are more dominant than others, the code that you receive is yours and yours alone, unless you have a twin. But genetics is something that is inherited from your family and passed down to you. Now, this code is going to be simply the genetic material, your code book, whereas epigenetics is going to be what we are here to discuss today, because epigenetics is the study of the biological mechanisms that actually help to turn genes on and turn genes off. Pretty cool, right? So looking at the top image, we have just a very simplified version of DNA. We have those base pairs that are put together and a very specific code, and that is just the genetic kind of makeup itself. Now, when we dive into epigenetics, we're going to look deeper at this in a minute, but epigenetics is the study of which of these genes are actually on versus which of these genes are off. Because fun fact, not all of your genes that you possess are on. Many of them are actually turned off, and that's a good thing. And we'll discuss why that is as we go through the slides today. So, epigenetics is the study of the heritable changes in gene expression. So your genes are not changing. Your code book is yours for life. That cannot change. But we can definitely see that things can be expressed in a different way. So remember, your genes are yours for life. You were born with them, you will die with them. And if you have children, you will pass some of that genetic material on to your children, your offspring. But in your lifetime, your genetic code will not change. What will alter and what will change is which of those genes are being expressed. And this is the study of epigenetics. So this is where we're looking at which of the phenotypes is being expressed, which of our genes are active, and which of our genes are actually inactive. And this can come down to simple things like whether or not we have brown hair or blonde hair or red hair or black hair, but this can also come down to whether or not genes that make us more likely to develop diabetes come into play or autoimmune conditions. So a big reminder, you and your genes are not going to change, but the expression of them, the phenotype that is expressed, which genes are turned on versus which genes are turned off, can change. This does not change the DNA sequence though. So this is very new science. The epigenome was actually discovered in the 1970s. And since then, the study of genetics and epigenetics has exploded to really help us dive into how we can help ourselves as far as understanding our own genes and how they can be expressed, but also learning more about what is it about our environment, about our lifestyle, about our traumas, about our food, about our interaction with others, about how we were raised? What is it about those things that actually affect how our genes are being expressed? This is the study of epigenetics. So epigenetics is going to be epigenetics affects how genes are read by the cells. So when this happens, when the gene is being read by the cell, depending on if that gene is turned on or turned off, the cell will subsequently decide whether or not it will or will not produce those proteins to make the substance at hand. And I'll make this make a little bit more sense in a second. So epigenetics is controlled through a couple of different things: nature and nurture. Epigenetics is not just when we're talking about, predisposing yourself to heart disease or to diabetes or to obesity. Epigenetics is also part of just you being human and being in utero and then coming into this world. So that's nature. Epigenetics is actually what determines a cell's particular function. So whether we are going to create a skin cell or a blood cell or a hair cell or a liver cell or any type of cell in the human body, that is determined by epigenetics. And as a fetus develops into a baby and then into a human, like a human adult, through gene expression, we will see which genes become active and which ones become dormant. So which ones are expressed and which ones are silenced. So even thinking about your own body, if we were to, let's say, take a piece of your hair and then take a piece of your skin, I know that sounds creepy, but just bear with me for a second, and we were to analyze the genetic material, both of those samples would provide you with the same genetic code, even though they look very different. Your hair sample and your skin sample have the same genetic code. But how is that, given the fact that the genetic book, that blueprint that we talked about, is the same, but they look so different? That's epigenetics. So as we are creating different cells throughout the body, we are kind of reading off this base blueprint, and then depending on what needs to be created, we can then express certain genes to make, let's say, an eye cell and suppress certain genes to not make a skin cell in your eye in the same way, or to make hair where there shouldn't be hair. So this is epigenetics, and this is also why in babies' eye colors can actually change up to about a year old. So you can be born with blue eyes, but then end up with brown eyes, or vice versa, or hair color can change as a baby ages. And this comes down to epigenetics. So part of this will also be the environment or nurture. So this brings us to the other way that we can actually see our epigenetic expression through not just the nature of, going through the world and starting to create that code and express that code, but also how our code, how our blueprint is interacting with the environment. And this is the nurture concept. So the environmental stimuli that we are exposed to can also turn genes on or turn genes off. And this is where epigenetics gets really interesting, because it turns out this can happen way before you were even born. It's a really interesting part of epigenetics to think about the fact that as you are in utero, so let's say while you are in your mother's womb, or when you were in your mother's womb, a pregnant woman's environment and her behaviors during pregnancy can actually change the baby's epigenetics. And some of these changes can actually remain in place for decades and can predispose someone to disease. But we can also reverse these in adulthood. Not all of them, but some. We see this not just in utero, but we see this also with what mom and dad are doing, or father and, or I guess just man and wife are doing, even around the time of conception and preconception. So these things can be passed down. So epigenetics is going far beyond the idea of, okay, this runs in my family, so therefore I am going to develop this too. We also now need to switch gears here and think about, okay, instead of just thinking that this runs in my family, I have cardiovascular disease, there's obesity in my family, etc. Now you have to ask another question. If you work with clients, this is a great one. What was food like growing up? What was your environment like growing up? What was movement and play like growing up? Because remember, yes, we pass down our genes to our next of kin, but we also typically pass on our experiences and our lifestyle. So this is where epigenetics becomes really interesting, because you are not only a product of your genes, you're actually mostly a product of your environment, especially in those childhood years. Okay, brief little pause for science here, and then we'll get back to the fun stuff. But how epigenetics works is through the concept of methylation. And methyl groups are everywhere in nature. If you've taken any type of science courses, or maybe you have taken our Certified Sports Performance course, we talk about methyl groups in our fatty acids, because there's fatty acids. Fatty acids contain a methyl group. There are other molecules that contain methyl groups that we'll talk about later on as we leave you with some tips and tricks to work with yourself and with clients. But the methyl group can be detached from certain things in our diet, and we can also make our own methyl groups as well, that we can then tag a gene with. So it's adding this to a gene, and when we add this methyl group, it actually suppresses the ability for that gene to be transcribed. So you can think about, by adding this tag, it turns off that gene, and in a good way. We want that to happen. We also see that histones can be modified, and histones are these proteins found in the nucleus, and they act kind of like spools that kind of help the DNA to wind around it. And DNA wrapped tightly around histones cannot be accessed by proteins that read the genes. And so these things are also quite helpful for either turning genes on and turning genes off. So why does this matter, and why is this so important? We'll talk about more of this in the food sense in slides to come, because we can actually increase the amount of methyl foods that we are consuming in our diets to help with this. But it's really important to take note of the fact that as we age, our ability to methylate decreases, and this can increase our risk of disease. So to give you an idea, When you are born, a newborn has the most amount of methylated genes, which means that less things are turned on, and we want that to happen. So DNA methylation at millions of sites were measured in this really cool study in newborns, in about a 25-year-old and someone who was over 100. And the level of DNA methylation was just shown to decrease with age. So the newborn had obviously the highest amount of DNA methylation. Many of these things were turned off and suppressed in a good way. But as we age, we saw that the older person, the person who was 100 years old, had the lowest level of methylation, meaning that many genes were actually active. And a lot of active genes can be problematic. Certain genes, when they are turned on, can predispose our risk of developing certain cancers or cardiovascular disease, etc. And we'll dive into this more when we talk about something like smoking. Okay. So to come back to that concept of this developing or beginning even before we are born, genetic expression can actually start before conception. So if we're looking at the developing germ cells in a fetus here on the top left corner, that can be passed down from family member to family member in generations to come. And these have been proven through studies, in particular around trauma. So one of the most famous studies around this was in the Dutch famine kind of age, that it was about 1944 to 1945, people whose mothers were pregnant during that famine time were more likely to develop certain diseases because of the fact that during conception and even preconception, mom, her genetic expression had started to change, and therefore that was passed down intergenerationally onto the next generation and then the next generation. So don't freak out. We're going to talk about how we can do this as adults and help ourselves in adulthood, but something to consider if you are working with people who are planning on conceiving as well. So one other thing here to mention is that both current and past environmental factors and dietary factors can influence epigenetic change. So what we eat now can affect us now, it can affect us later, and if we have kids later on in life, this can affect them as well. So one of the most interesting studies on this was done in mice, and this was done looking at a very particular gene called the Agouti gene, and mammals have this gene, so we have this gene too. And the study was quite interesting because when a mouse's Agouti gene is completely unmethylated, so that means that little tag is not there, that methyl group is not attached to that gene, the coat of this mouse turns yellow, and it's actually obese, and it's prone to diabetes and cancer. But in a mouse that is exactly the same, because in this type of testing, they clone all the mice. So these two mice would have the exact same genetic code, but the yellow mouse here has an unmethylated Agouti gene. The brown mouse has a methylated gene, so it's a normal mouse. And the coat is brown, and the mouse has a very low risk of disease in comparison to that yellow mouse with the unmethylated gene, who has an increased risk of diabetes and cancer. So what does this mean for future generations? In this study, it was really interesting to take note of that fat yellow mice and skinny brown mice were identical. They were genetically the exact same in this test. And because of the unmethylated gene, that's just why it was obese. Okay. Now, in this study, when researchers fed pregnant yellow mice, so that means that they were pregnant, but they had this unmethylated gene, they fed these mice methyl-rich diets. And we'll talk about methyl donors as we come to the end of this lecture today. When this pregnant yellow mouse gave birth, most of her pups were actually brown, and they stayed healthy for life. So just going to show that what we eat in utero and what we are exposed to in utero really matters. And we can also see that this can change in adulthood as well. We'll discuss this later on. So these results suggest that health is not only determined by what we eat and what we experience, but also what our parents experience. And because we can't go back in time, I promise I'll spend a lot of time at the end here talking about how we can help ourselves as adults. but it is something just to make mention of here, and it's quite worth noting. Okay, so on that note, epigenetic changes occur at different rates at different stages of life. And while we see most of the most prevalent and kind of fastest and most prominent changes in childhood and in utero, we can see changes in adulthood. And exposure to certain environments and foods and chemicals or no chemicals or stress, and we'll dive into all of this, can actually alter which genes are exposed. And this can happen if we're doing this prenatally, but also later on in life as well. When this happens in childhood, however, or even prenatally, the effects are going to be more prominent, or they are going to be more long-lasting and life-changing, because we can actually see these things happen quite quickly. But if we're thinking about this from, okay, right now, what can we do? And how can we help ourselves? And how could we help our clients with our own gene expression in adulthood? Here's how we're going to do this. While epigenetic markers are more stable as adults, they can be altered and they can be influenced. And this comes down to not only the food that we eat, but the lifestyle that we have and the environmental exposures that we are exposed to. And that in itself is a big can of worms. So let's start with smoking. It's a really interesting study here on smoking with people who were long-term smokers, and their methylation changed after they quit. So obviously we know that smoking is not good for you in any way, and smoking can actually result in changes to your epigenome. So certain genes are actually going to be expressed differently when you are a smoker. So looking at one specific gene, and we're just going to keep it here as a small little cap, as the AHRR gene, smokers tend to have that gene be far less methylated than non-smokers in studies. And the difference is definitely going to be greater for heavy smokers and those who have smoked long term. But in studies, after these people quit smoking, former smokers actually began to have increased DNA methylation on this gene. So we saw that that gene actually became suppressed in a good way. We started to methylate it and turn it off. And eventually, many of these people actually reached similar levels of methylation on this gene to non-smokers after changing their lifestyle and stopping smoking. So something just to show us that even as we age, we can turn certain genes off and we can change our phenotype. We can change which genes are being expressed. Now, things like eye color, it's not going to change, but there are certain things like, let's say, slowing down the rate of gray hairs, working to suppress autoimmune conditions. All of these things can be possible so long as we are thinking about all of these factors at hand. So things that we can think about that can affect our epigenetic expressions. Smoking and drinking for sure. So if you have clients who are excessive smokers or drinkers, definitely the easiest low-hanging fruit is to start to reduce the amount of exposure to these things, because they can change our ability to methylate properly. Also, these things will cause more stress on your liver and stress on your detoxification systems that use methylation for detoxification purposes as well. Methylation happens not just at a genetic level, it also happens in our detoxification pathways. We use the methyl groups to actually kind of make things less toxic as we're removing them or getting ready to remove them. Another big factor is stress. If you've ever maybe met up with a friend after many years and they've had a very stressful life, they look different. Or if you've seen twin studies where one person was stressed and one person was not, they look different, even though they're actually the exact same genetically and they're the same age. So stress can affect our ability to methylate properly. It can also affect antioxidant status, that can have other effects that can then affect DNA methylation. So reducing stress through meditation, through just letting go of some of the things that might be causing them too much stress, maybe not driving in traffic, all of these things can be really useful things that can help us to change our epigenetic expression. Not listed on the slide is sleep, but I would put sleep of utmost importance on this as well, because if we are not getting adequate sleep, this can also affect our epigenetic markers just because it is a stressor. Big ones to consider are environmental exposures, and this in itself could be a very interesting webinar topic, but the pollution that we're exposed to, the air quality that we are breathing in on a regular basis, the toxins that we have in our hair care products, our water, our plastic containers, the makeup that we might be wearing, the clothes that we buy, the furniture that we sit on, those things can actually affect our epigenetic expression as well. Just simply because those chemicals can alter our genetic function and can also cause a just influx of free radical damage, because some of those things are what we call forever chemicals, and they're quite hard to get rid of in the body. So reducing your exposure through maybe diving into the realm of non-toxic healthcare products, non-toxic cleaning products, beauty products, you name it, can actually be another way to reduce your kind of exposure to these things and change your epigenetic expression. I know that seems far-fetched for some, or it might. Maybe for some of you that sounds quite normal and you've already gone down that path. But this is a huge topic of discussion that is really coming to the surface in the last year or two to dive into more reconsiderations around environmental exposures and our overall health. So a big thing to consider there. This is not your low-hanging fruit with clients. This is definitely not something that we would be diving into on the first bat, simply because there are other lower-hanging fruits like the diet and like, let's say, reducing stress and reducing smoking and drinking. But other things to consider for long-term health would definitely be looking at those environmental exposures and thinking, okay, how can we alter this? How can we reduce that toxic load for this person? And if you don't know how to do this yourself, please do reach out to someone in my field or in the field of naturopathic medicine as well, because this is something that we do quite a bit of work of, and we see this being not the low-hanging fruit, but the kind of longer-term project management as far as thinking about someone's overall health. And dietary choices. Now, there are lots of dietary choices and kind of things that we can consider with diet and epigenetics. And this is why I really suggest adding this question to an intake form or even discussing this with your clients, because it's not just for the epigenetic purpose, but also thinking about how well they fare with, let's say, eating a more whole foods based diet versus a more processed food diet. I would think, okay, what was food like growing up for you? Did you eat lots of home cooked meals? Were you eating lots of processed food? Was there lots of takeout? And really diving into what their experience was with food and letting them talk about it too. And then if you have more questions, go forward. But diet is very well studied for its effect on epigenetic change. Remember that diet is more than just the calories that we consume. It's even more than the macronutrients that we consume, because the nutrients that we extract from our food actually enter metabolic pathways where they are manipulated and modified and molded into molecules that our body can use. So beyond just getting energy in the form of ATP from our fats and our proteins and our carbohydrates, we also will be obtaining, or should be obtaining, decent amounts of antioxidants, decent amount of micronutrients, so in the forms of vitamins and minerals, that play important roles in kind of balancing the amount of oxidative stress that we're under, so kind of making sure that we have adequate antioxidants for the amount of free radicals that we have, but also helping us to make sure that we have methylation pathways open. So in the case of epigenetics, we are going to be looking more at the methyl-making foods, but for overall health and thinking back to epigenetics and like more of a grander scale of things, if you are eating poor quality food, this can also affect your epigenetics, not just from the lack of folic acid and B vitamins and SAM-E, but also from the high amount of free radical damage and lack of antioxidants that your body is, one, not making, and two, not eating to fight those free radicals, that can then also affect your epigenetics, can affect your liver function, can affect all sorts of things, is how that cell is being expressed if we are not getting adequate nutrition in. So the main methyl donating foods, or the methyl making foods, are going to be the B vitamins, actually. So B vitamins in general, but there are quite a few that are methyl donors. So folic acid being one of them, B12 is another one. And then you have a few that are actually just quite protective for your DNA, biotin being one of those. SAMe is another component that we actually can make from some of our micronutrients to then also create methyl groups to donate and have that be part of this methyl making pathway. So it's very useful for liver function as well as for your epigenetics. And more research proves that diets that are high in these methyl donating nutrients can actually rapidly alter gene expression, in particular during early development when the epigenome is first being established. So this little human is starting to express certain genes and suppress certain genes. It's much more potent then as far as like what it's going to affect for that human who is changing. But even in adulthood, when we have a diet that is rich in these nutrients, in these methyl donating nutrients, that's also rich in antioxidants, that's rich in just good quality nutrition and is not full of chemicals that are put on the food, not full of, let's say, damaged fats and damaged sugars and all of those things that we don't want to be eating, then the body will be in a better place to support a healthy epigenetic expression. So we can think about obviously extracting these nutrients. We as nutritionists and even as personal trainers who work in the realm of working with nutrition suggestions tend to sometimes go into the realm of, okay, what's this person's macros, what are their micros like, and kind of thinking about what they need on the plate. But another way that we can also kind of think about what this person needs is, okay, aside from getting energy from food and making sure that you have vitamins and minerals, we also need to think that they do other things in the body. The micronutrients are absolutely incredible, and we dive into that quite deeply in the Certified Sports Performance and Fitness Nutrition Specialist course, but they do a slew of things for us aside from helping us to metabolize nutrients. Some of them are useful for muscle relaxation, contraction, all sorts of fun stuff. But in the case of things like choline and B vitamins and methionine even, which is quite important for making sure that we don't have high levels of like a not so great amino acid in our body. But these things can be extracted from food, and the folate, which is B9, the B12 are the two ones that I'll just kind of make mention of right now, and even choline, they are methyl donors. So that little picture that I showed you at the beginning of that carbon with 3 hydrogens, that can actually be detached from these nutrients totally safely and then attached to our literal DNA, which is amazing. And that can then turn not so great genes off and suppress that. So eating a diet that is rich in these nutrients is quite important for our overall health, for detoxification, and also for our epigenetic expression. And no, you cannot just supplement these and continue to eat poorly. We do need the other nutrients that kind of work in the background to make all these mechanisms run. But in some cases, supplementing on top of a good diet could be useful. So if we are thinking about some of these nutrients, if we're looking at methionine, which is an amino acid from food origin, obviously we're going to be finding this in most just complete proteins will have a decent supply of methionine, but you do find it in higher amounts in sesame seeds, Brazil nuts, fish, and peppers and spinach. Now, its kind of reason or role, is thinking about SAMe synthesis or SAM synthesis. Folic acid. Folic acid is technically the lab-made version of it. You'd be looking more for folate in food, and then in the body, we convert this into its active form called 5-MTHF. In food, we find this in liver, we find this in sunflower seeds, in baker's yeast, and dark leafy vegetables. And it helps us to create methionine. So it plays a role in creating more methionine in the body, because fun fact, a lot of amino acids, unless they are and some of them we can actually make from other amino acids. So we have our essential amino acids that we need to obtain from the diet, and then we have others that we can actually make. So methionine we can make from other nutrients, or play a role in how we make this specific amino acid. B12, B12 is only found in animal products. So the B12 that you find in plant-based products are actually a pseudo B12 that do not do the same thing as B12 in animal products, because the B12 that we find in animal products is going to be from like microorganisms that grow and kind of compound in animals, and then we eat those animals. So in meat, liver, shellfish, and milk, you will find higher amounts of B12, and this is very important for methionine synthesis. So for clients who are plant-based and vegetarian even, ensuring that they get their B12 levels checked and then supplementing accordingly is definitely recommended. B6 is found in meat, whole grains, vegetables, and nuts, also important for methionine synthesis. Sulforaphane is found in broccoli, and you can actually increase your sulforaphane content by chopping your broccoli a little bit before you use it. That can help to increase the amount of this molecule that is found in broccoli. So it's a sulfur-containing compound, and it helps to increase the histone-acetylation. So it helps to turn on anti-cancer genes. So while obviously this is just more of a preventative kind of role, that compound is extremely helpful for working to turn on anti-cancer genes and turn off the ones that can kind of potentially mutate. Right? And butyrate. Butyrate is a really cool compound because it's actually produced in the intestine when dietary fiber is fermented. So it's actually made from gut bugs, but you can also find butyric acid in foods like butter. So grass-fed butter can also get you butyric acid or butyrate, and that can be very helpful for increasing histone acetylation as well. Having adequate levels of butyric acid and butyrate can be shown to increase lifespan. This wasn't done in flies, but even as a compound, it's a very healthy compound to have in the human body. It's also used as a fuel source for the brain, which is interesting. Okay. So now thinking about how we can actually move forward with this information. Now that you know this, it's pretty fun and it's pretty interesting to think, okay, everything that I do in a day can affect my genetics, but it can also affect just literally my body all the time. I know that this is genetic specific, but it comes down to, you know, affecting all of your cells all the time. But we really want to think about creating a gene supportive environment. And when we create a gene supportive environment, this means that we are working to create the best versions of ourselves from the inside out and from the outside in, right? So from what we're eating coming in, and also from, you know, the toxic exposures that we might be exposed to, all of that. So that way our genes from the inside out can be expressed in a very healthy way. So things to consider when creating a gene supportive environment. Thinking about the macronutrient ratio can actually be a good way to do this. And I'm not saying that there's one perfect macronutrient ratio, but we're obviously thinking about having adequate levels of high quality protein, having healthy levels of quality fat, because I find a lot of people are still eating very low fat diets. We do need a healthy amount of fat on your plate as well, and carbohydrates. And quality of carbohydrates really, really matters. We want to make sure that we're getting adequate quality whole food carbs to obtain many of those methyl donating nutrients. With the macronutrient ratio, this will really vary. I'd say about 80% of the population can really work with just like a pretty even macronutrient ratio. But on either end of that spectrum, there are some individuals, about 10% on either side of that macronutrient ratio. Some will fare better with a high fat, medium protein, low carb diet, and then the others will fare much, much, much better with a high carb, medium protein, low fat diet. And to understand that, you do need to go for genetic testing to understand if you are one of those people who does fare better, let's say with less fat or less saturated fat, or with way more saturated fat for your health, because yes, some people actually need more of those nutrients to have a gene supportive environment. But in a nutshell, for the average person, just kind of sticking to a pretty balanced macronutrient ratio is a pretty good place to start. But also thinking about the quality of food. We really want to steer clear as much as possible from the amount of packaged food that is out there, the amount of kind of processed vegetable oils, processed sugars, just anything that is chemically laden is definitely something that we want to avoid. And when working with, let's say proteins, getting your animal proteins from grass-fed sources whenever possible, pastured sources, and if you're working with plant-based proteins, obviously making sure you're getting your whole slew of amino acids and being mindful that you do need a little bit more protein just to increase that, because your bioavailability is slightly lower, and also making sure that you're not just eating more of the processed plant-based meats too. Amount of food also matters. So thinking about the amount of food that you eat in a day, overconsumption can also affect epigenetics. So thinking about getting the right amount for you, and this will obviously vary from person to person, their exercise levels, etc. But overeating can also be a problem. So this typically is remedied for most people when we adjust the macronutrient ratio, and when we adjust the quality of food, we tend to find a natural intake that makes sense and feels satiating for that person. But also thinking the other end of the spectrum, if you are not eating enough, this can be a stressor to the human body. This can change cortisol secretions. This can change stress hormones to the point where this can also alter your epigenetics. And this was proven through that study in the famine in the Netherlands back in the 1940s. Timing of food and also kind of if you are fasting or not fasting, or if you're constantly eating. And this gets a little bit more intricate, but fasting for some people can actually be a really useful part of epigenetic health. And for others, it might be more of a stressor. So it will really be thinking more about this person and who's in front of you and what's right for them. But on the other end of the scale, if you're constantly eating, if you're staying up late at night, this can also affect digestion, and therefore down the line can affect other aspects. So in a nutshell, thinking about food starts with that macronutrient ratio. I find that's the easiest low-hanging fruit, is to kind of alter that and then jump into the quality of food, and the rest of it kind of flows as you see fit. Okay, and then lifestyle. So creating that gene supportive environment goes beyond just thinking about what's on your plate. And while that does play a major role, we also have to think about what else our cells are exposed to on a regular basis. And yes, this means stress. When you are stressed, your body does not know the difference if that stressor is coming from a really great workout, or you being stuck in traffic, or you getting into a fight. Your cellular response is the exact same. So someone's bucket of stress, someone's allostatic load is really important to take into consideration. So stress management is a big part of that plan. If someone is in a super stressed state and they're working out 6 to 7 days a week, it might actually be wise for them to cut back on their training so they have 2 to 3 great workouts that are intense but awesome, so they can come back to baseline in between, because this can actually create a better gene supportive environment, and they might also see better gains out of it too. Quitting smoking, definitely hands down, one of the better things you can do is encourage your clients to quit smoking if they still are smoking, just for long-term health and for all the other health benefits of quitting smoking. Limiting alcohol consumption, there's an H in there that should not be in there, but limiting alcohol is also important, just for the fact that this can affect liver function. We know it is a carcinogen, but being mindful that this might not be the low-hanging fruit for everyone, and not everyone will be ready to hear that. And movement, you have to keep moving. So when we look at movement being part of a gene supportive environment, we know that exercise can stimulate all sorts of fun things in our immune system, in our stress response in a good way. All sorts of fun stuff comes from moving on a regular basis. So as we age, or even in our younger years, we do want to make sure that we have healthy movement, and we're not being stagnant or sedentary for too long, just simply because we are designed to be moving as human beings, even though our modern lifestyle is really encouraging us to sit and not move and order food as much as possible. So going against that can be quite helpful for your long-term health and also for your short-term health. So through these things, we really wanted to think about the fact that this is not just for the long-term goals. This can also affect someone's short-term goals. So I will jump into that as we kind of go into our summary here. But a little reminder for yourselves that when it comes to most conditions, when it comes to most illnesses out there, most kind of conditions that run in families, we are very quick to think about the fact that, oh, it runs in my family, so it's inevitable that I'm going to get it. But for most of these conditions, it's about 20% genetics. So 20% in that code book that you've inherited. The rest is your environment. The rest is what you're exposed to every single day. And while, yes, a lot of that has to do with childhood, what has changed in adulthood and your exposure in adulthood also matters. Thinking back to that smoking gene for a second, 80% is this person's environment or your environment. So thinking about the food you eat in a day, thinking about the movement that you do, thinking about the ways that you cope with stress and manage stress, thinking about the quality of food, the environmental exposures that you might be able to limit for yourself, all of these things can help you to create a gene supportive environment. And the more you dive in for this for yourself, the easier it will be to kind of share this information with clients as well. So in summary, we discussed the idea of genetics. We then explained epigenetics and in a nutshell and how epigenetics affects us. And we discussed the idea of creating a gene supportive environment. And while I did spend quite a bit of time talking about this from the long-term perspective of, okay, let's make sure that we don't predispose ourselves to chronic illnesses or cancers, etc. This also works in the short term as well, as far as setting our bodies up for success for short-term goals. Even though our genetic marker, our epigenetic markers won't magically shift overnight, this is something that takes a long time. We can also think about it from the preventative point of view of, okay, this client will be healthier for longer. They will have less likelihood of developing certain things if we do this now. But also going back to these things, we're definitely going to be seeing short-term changes if we're able to alter these things as far as nutrients go on their plate, and we're able to alter these things to create a less stressful environment, to have great exercise plans, to have great recovery plans in a way that's going to have them feeling good now and see changes to let's say whatever goals that they're looking at, but also to support that client maybe even beyond their time with you, maybe into the next 10, 20, 30, 40 years of their life, if we are able to think, okay, how can we create a gene supportive environment? So epigenetics in a nutshell, remember that 20% is genetics, the rest is your environment. And I hope that you found that interesting. I hope that that sparked something inside of you. And if you are curious about learning more about epigenetics and how you can work with epigenetics, there are companies that as personal trainers you can actually dive into. But I will open the floor to questions and see if anybody has anything that has come up through this webinar this evening.
Thanks, Sydney. That was a bunch of really fantastic information. We do have a few questions. The first one being, what is SAMe?
Okay, so SAMe is a molecule that we can produce in the human body. You can also find it in supplement form. And when it comes to this molecule, the supplement, if you've been ever looking this up, is typically something that is discussed for depression or for kind of elevating mood. But SAMe is actually it's a molecule that we make, and it helps to control levels of homocysteine in the body. So we make this internally from homocysteine, which is a good thing. We can take it and make less homocysteine. We also will use folate to make this. And with this molecule, it helps us to break down chemicals. It helps us to work with proteins and break them down in a good way, break down old hormones, old chemicals, and can also actually break down drugs and make them less harmful in the body. So it's useful for liver function, it's useful for our epigenetic expression, and it also plays a role in pain management and in depression when we use it as a supplement. But we do make it internally. So that's what SAMe is. It has a very long name, but we're just going to leave it at that.
Awesome, thank you. Next question is sulforaphane only found within broccoli?
Very good question. Off the top of my head, it's the highest amount in broccoli. The sulfur containing compounds are found mostly in the brassica family vegetables, but with sulforaphane, it is mostly found in broccoli. It will also be found in cauliflower in smaller amounts, and in Brussels sprouts as well.
Awesome. What is homocysteine?
Okay, so homocysteine is it's an amino acid that is not so great for us. So homocysteine is something that we do make internally, and it's part of it's part of kind of like a detoxification process where we need certain amounts of homocysteine. But when they get too high, this can kind of raise our likelihood of developing certain illnesses. And this kind of balance of having adequate levels of homocysteine, but not too much, comes down to having decent amounts of folic acid, or sorry, B9 or folate, and B12 in the body to help us convert homocysteine to methionine, because we need methionine. We use a lot of methionine, but we don't want to have too much homocysteine. But in a nutshell, it's actually an amino acid. And
Great.
high levels of, sorry, one more thing on that. High levels of homocysteine are correlated to high levels of disease, as far as like cancers, inflammation, inflammatory diseases, you name it. Having a buildup of homocysteine is typically a sign that you're not detoxifying properly. You don't have enough of those vitamins, and your liver might not be able to convert homocysteine to methionine properly.
Awesome.
Mm-hmm.
Next question, is there any relation between the chromosome telomeres, epigenetics, and methylation?
Wow, that's a crazy detailed question. That goes beyond my knowledge of epigenetics, and I will not try and make up an answer for that, because that's something that I simply cannot think of off the top of my head. With the telomeres and epigenetics and methylation. Interesting though, and I will be diving into that question on my own time after this is over this evening, but I'm sorry I don't have an answer for you.
How long does it take after you quit smoking to see positive results in your epigenetics?
Ooh, good question. The studies that I was looking at, some showed it within like about 6 months to a year period, but others it showed longer. And the biggest thing that was mentioned was just it depends on how long you smoked for, and it depends on how heavy a smoker you were, and also other factors in your life as far as your antioxidant status, your stress levels. Other lifestyle factors will play a role in that as well, because we have to think about the fact that nothing in the human body is black and white, and nothing is separate. So everything that you do in a day can affect that as well. So there's no magic kind of specific answer for that, but it will depend on all of those factors, which is why creating that gene supportive environment is so important.
Great. Where can you go for genetic testing, and what tests would you suggest?
Oh, good question. So there's actually a wonderful company that's located in Vancouver that you as a personal trainer can actually access, and you can do it for yourself, and you can even learn more about it in your own practice if you wanted to. It's called DNA Power. And so that's a cool test to run for yourself. In those tests, you can kind of choose from a suite of tests that can just focus, let's say, on nutrition aspects. Some will focus on brain health, or you can kind of buy all of them together. They're not cheap, but remember, these are just going to be looking at your genetics, not your epigenetics. So the genetic tests that are available on the market for just anybody to purchase are going to be looking at the genes themselves, not the epigenetic expression. So that would be, going into crazy lab work to do stuff like that most people will not have access to. But understanding your genes and thinking about, okay, this is in my genetic code, maybe I should be more mindful of how I eat, or whether or not I decide to smoke, or whether or not I decide to drink, because on my genes, even though I don't know if it's being expressed or not, this is something I am predisposed to. So taking tests like that can be quite helpful if that's something you wanted to dive into. So DNA Power is one of them. There are actually a couple of supplement companies that will, and I don't want to name names for that on here just because I don't know if we can or not, that offer a panel, like a genetic testing done through their company, and then they will suggest obviously supplements from their brand to line up with the things that have come back on your genetic test.
Without getting testing done, how would you know if fasting is good for you?
Oh, that's a great topic that we could do a whole chat on that. But fasting is one of those things that for some people it can be extremely helpful. For others, it can be detrimental. In particular, for females of menstruating age, it tends to be something that is more of a stressor, simply because women and female hormones, anyone who has female hormones, will be more likely to be prone to like the stress response of not eating for long periods of time. But even those who are who have male hormones can not always benefit from fasting. So signs that fasting is not working for you is that you are shaky, you're stressed, you feel cold, you're losing gains. There's many different factors that can kind of help guide you, and you have low libido, low energy. These things might not be right for you as far as fasting goes. But with fasting for males or females, you always want to go into it very slowly. So like 1 day a week of like pushing your breakfast 1 or 2 hours, and then the next week, same thing, 1 day a week, pushing it a few more hours, really kind of just helping you, kind of like training in the sense of expanding your allostatic load and kind of like getting you used to that stressor in a way that doesn't seem so extreme. So don't just jump into a 24-hour fast or anything like that, because it will be more of a stressor than good. It's the same thing as, you know, you going to a bar and lifting up 250 pounds in a deadlift and you've never done it before, you're going to hurt yourself. So with fasting, go slow and kind of wait for those cues as well.
And one last question for this evening. Could you provide a quick recap of all the vitamins, molecules that go into affecting epigenetics?
Oh, okay. There are lots of different molecules. I didn't even name them all. But if we go back through these slides, these are the main ones that affect epigenetics. So folic acid or folate coming from food, folic acid is supplement form, and you really want to not find folic acid. You want to find the active form if you're buying a supplement called 5-MTHF. But the B9 and B12 are extremely important. B6 as well plays a big role in methylation and detoxification, which plays a role in our ability to express proper epigenetics as well. So the B vitamins are the biggest ones. It's like the low hanging fruit for epigenetics would be ensuring you have adequate levels of B vitamins, and then going into compounds like antioxidants and sulforaphane, but also just other antioxidants to help reduce inflammation. And then looking at the other ones on this list. But the B vitamins are the first ones I would discuss. Yeah.
Wonderful. Thank you so much for all this fabulous information that you provided us with this evening.
You're so welcome.
And thank you for everybody that listened in tonight. We also appreciate all of you. Sidney, do you have any final comments?
Always, always, always make sure that when you are creating plans for yourself and for your clients, that everyone is different as far as their ability to make change, their desire to make change, and also even what a gene supportive environment looks like for them. So checking in with your clients and making sure that they're ready to hear this information. If you are going, to the gym tomorrow or going to work tomorrow, just check in to see where they're at as far as their next steps go, and see what you can implement that can help to create that environment for them, but in a way that's going to add in to their current lifestyle in a positive way. That's always my my kind of 2 cents and final goodbye there. So I hope that that's helpful for you.
Fantastic. And if you'd like to learn more information, as Sidney has spoken of, we do have a certified sports performance and fitness nutritionist specialist program that Sidney did help create. So you can contact us and we'll be happy to give you more information about the program. And I'd just like to say good night to everybody. Good night, Sidney, and hope all of you have a wonderful evening.